So chapter number 9, biomolecules. So context are how to analyze chemical composition, primary and secondary metabolites, right? Biomacromolecules, then proteins, then polysaccharides, then nucleic acids, then structure of proteins, and last is the enzymes, right? So are all living organisms made up of the same chemicals, that is elements and compounds? Okay. If the same analysis is performed on a piece of Earth's crust as an example of non-living matter, we obtain a similar list. So living tissue and Earth's crust tissues, Earth's crust, the the elemental analysis is almost the same, right? No such differences could be made out. All the elements present in a sample of Earth crust are also present in a sample of living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to other elements is higher in any living organism than in the Earth's crust. Right? Now, how to analyze chemical composition? There is trichloroacetic acid using a mortar and pestle. There is the formation of slurry. One is called the okay. Then when we filter it out, one is called the filtrate, or more technically the acid soluble pool, that is dissolved, and the second the retentate, or the acid insoluble fraction. The scientists have found thousands of organic compounds in the acid soluble pool, right? All the carbon compounds that we get from living tissues can be called biomolecules. So all the carbon compounds in the living tissue are called biomolecules. However, living organisms have also got inorganic living organisms, elements and compounds in them. How do we know this? Now, if the tissue is fully burned, all the carbon compounds are oxidized to gaseous form, CO2 and water vapor, and are removed. And what is remaining is called the ash. Now, this ash contains inorganic elements like calcium, magnesium. Inorganic compounds like sulfate, phosphate are also seen in the acid soluble fraction. So therefore, elemental analysis gives elemental composition of living tissues in the form of hydrogen, oxygen, chlorine, carbon, while analysis for compounds gives an idea of what kind of organic and inorganic constitutes present in living tissues. Now, amino acids are organic compounds containing an amino group and an acidic group as substituents on the same carbon, that is alpha carbon. Hence, they are called the alpha amino acids because on the same carbon they have the amino group and an acidic group. Now, they are substituted methanes. So, amino acids are substituted methanes. There are four substituted groups occupying the four valency positions. These are hydrogen, carboxylic group, amino group, and a variable group designated as R group. Based on the nature of R group, there are many amino acids. However, those which occur in proteins are only of 20 types. The R group in these proteinaceous amino acids are proteinaceous, could be hydrogen. The amino acid is called the glycine. When it is a methyl group, that is alanine, and hydroxyl methyl, that is serine. So 3 of the 20 amino acids are given in your NCERT. The chemical and physical properties of amino acids are essentially of the amino, carboxyl, and the R functional groups. Based on the number of amino and carboxyl groups, there are acidic. Based on number of amino and carboxyl groups, there are acidic, that is glutamic acid, and basic, that is lysine, and neutral, that is valine amino acids. Similarly, there are aromatic amino acids: tyrosine, phenylalanine, and tryptophan. A particular property of amino acids is the ionizable nature of NH2 and COOH groups. Hence, in solutions of different pH, the structure of amino acids changes. Because amino acids has a particular property which is the ionizable nature of NH2 and COOH. Hence, in solutions of different pH, the structure of amino acids changes. As you can see, first diagram NH3 positive COOH, in second diagram NH3 positive COO negative, right? Both in the ionized form, and then third is the NH2 COO negative. Okay, B is called the zwitteronic form, right? Now the lipids are generally water insoluble. Lipids are generally water insoluble because they are the association of membranes, vesicles. They could be simple fatty acids. Now a fatty acid has a carbonyl group attached to an R group. So fatty acid has a carboxyl group attached to an R group. The R group could be methyl, ethyl, and a higher number of CH2 groups, that is 1 to 90 carbons. Example, palmitic acid 60 carbons including carboxyl carbon. Arachidonic acid 20 carbons, 20 carbon atoms including carboxyl carbon. Now fatty acids could be saturated, that is without double bond, or unsaturated with one or more C double bonds. Okay, so saturated without double bond, unsaturated with one or more carbon double bond. Another simple lipid is glycerol, which is trihydroxypropane. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. As I told you, lipids could be simple fatty acids, and glycerol is also a simple lipid, which is a trihydroxypropane. But many lipids, they have both glycerol and fatty acids. Here the fatty acids are found esterified with glycerol. They can be then monoglycerides, diglycerides, and triglycerides. These are also called fats and oils. So when they are esterified together, this can also be called as fats and oils. Now the lipids can also be called as fats and oils based on melting points. Now oils have lower melting point, that is jingly oil, and hence they remain as oils in winters. You can also identify a fat from the market with this method. Some lipids have phosphorus and a phosphorylated organic compound in them. Okay, these are phospholipids. Now they found in cell membrane. Lecithin is one example, one of the phospholipid. Some tissues, especially the neural tissue, have lipids with more complex structures, right? Now living organisms have a number of carbon compounds in which heterocyclic rings can be found. Some of these are nitrogenous bases: adenine, guanine, cytosine, uracil, and thymine. So the nitrogenous bases adenine, guanine, cytosine, uracil, and thymine, when found attached to a sugar, they are called nucleosides. When these nitrogenous bases found attached to the sugar, they are called nucleosides. If a phosphate group is also found esterified to the sugar, they are called nucleotides. Now adenosine, guanosine, thymidine, uridine, cytidine, and are the nucleosides. Adenine, guanine, thymidine, uridine, and cytidine, these are the nucleosides. Adenylic acid, thymidyllic acid, guanidinic acid, uridylic acid, cytidyllic acid, these are the nucleotides. Now nucleic acids like DNA and RNA consist of nucleotides only. DNA and RNA function as genetic material. So as you can see, the glycine H, CH3 alanine, serine, CH2OH, hydroxyl, methyl, hydroxyl methane, hydroxyl methyl. Okay, as you can see, hydroxymethyl in serine, right? These are the amino acids. Now these are the sugars that are carbohydrates. C6H12O6, this is glucose. C5H12O5, that is ribose. OH downside. one OH is upside at the third position in glucose, and rest are downward. OH, CH2OH. So these are the sugars and carbohydrates. Now fatty acid, that is palmitic acid, including 60 carbon atom, then glycerol, then triglyceride. You can see that three R1, R2 and R3. R1, R2 and R3 are fatty acids, and in the middle that is a glycerol. Okay, and what are the fatty acids? They have the carboxyl group, and they have the carboxyl group, and they have the R group. Okay, it can be like methyl, ethyl. So these are the fatty acids. Then this one is the glycerol. It is a simple lipid, trihydroxypropane. Then phospholipid, that is a lecithin, found in cell membrane. You can see R1, R2, and there is a phosphate compound, and then N, CH3, CH3, CH3. So this is a phospholipid. Remember the structure. This one is cholesterol, OH on this side. 1, 2, 3, 4, 4 rings are present in cholesterol. These are fats and oils. All these are lipids, right? All these are lipids. Now another thing is adenine, that is purine. Then adenosine, that is nucleoside, and then adenalic acid, that is nucleotide. As you can see, adenine, purine, 4 nitrogen group in the 2 rings. Overall it is 5 rings, 5 nitrogen atoms. Then adenine is attached to the sugar. Okay, adenine is attached to the sugar, and then adenalic acid, you can see phosphate group is attached to the sugar. It is nucleotide. Now uracil, it is a pyrimidine. Adenine and guanine, purine, uracil, cytosine and thymine, these are pyrimidine. 1 heterocyclic ring, 2 nitrogen present in the ring, and then there are nucleosides, that is uridine, uracil attached to the pentose sugar, and then there is a nucleotide. So all these are diagrammatic representation of small molecular weight organic compounds in living tissues. Now primary and secondary metabolites. Right? So biomolecules, we call it as a metabolites in animal tissues. Okay, so there are primary metabolites. However, one analyzes plants, fungal, see thousands of other. Okay, so there are primary metabolites in animals, right? However, when one analyzes plant, fungal, microbial cells, one would see thousands of compounds other than these primary metabolites. Okay, so examples are alkaloids, flavonoids, rubber, essential oils, and antibiotics, color, pigments, scents, gums, spices. These are secondary metabolites. I repeat it again. Other than the primary metabolites, there are alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums and spices. These are secondary metabolites. Okay. Now when primary metabolites have identifiable functions, they play roles in normal physiological processes that we do not know at the moment. Understand the role or functions of all the secondary metabolites in host organisms. However, many of them are useful to human welfare, that is rubber, drugs, spices, scents and pigments. Some secondary metabolites have ecological importance. In later chapters, you will learn that. Okay, so these are the secondary metabolites that do not have the an identifier functions like the primary metabolites do have, and they play a role in normal physiological processes. Now the secondary metabolites are given in the NCERT. These are pigments, alkaloids, terpenoids, essential oils, toxins, lectins, drugs, and polymeric substances. Now, pigments. These are carotenoids and anthocyanins, alkaloid. These are morphine and codeine. Terpenoids, monoterpenes, diterpenes, essential oils, lemongrass oil, etc. Toxins, urbine and ricin, lectins, concanavalin A, drugs, vinblastine and curcumin, and then polymeric substances, rubber, gums, and cellulose. So these are the secondary metabolites. Now, so we are going to study the biomacromolecules. Now, there is one feature common to all those compounds found in the acid soluble pool. They have molecular weights ranging from 18 to around 800 daltons approximately. So 18 to 800 daltons approximately in the acid soluble pool. The acid insoluble fraction has only 4 types of organic compounds: the proteins, nucleic acids, and polysaccharides and lipids. These classes of compounds, with the exception of lipids, have molecular weights in the range of 10,000 daltons and above. From this very reason, biomolecules, that is chemical compounds found in living organisms, are of two types. One, those which have molecular weights less than 1000 dalton, that is 80 to 800 dalton, and are usually referred to as micromolecules or simply biomolecules. These are thousand in number, while those which are found in the acid insoluble fraction are called macromolecules or biomacromolecules. Right? The molecules in the insoluble fraction, with the exception of lipids, are polymeric substances. The molecules in the insoluble fraction, with the exception of lipids, are polymeric substances. Then why do lipids, whose molecular weights do not exceed 800 dalton, come under acid insoluble fraction, that is macromolecule fraction? The lipids are indeed small molecular weight compounds and are present not only as such but also arranged into structures like cell membranes. So lipids, they form the cell membrane and other membranes. So when we grind a tissue, we are disrupting the cell structure. The cell membrane and other membranes are broken into species and they form the vesicles, which are not water soluble. So therefore these membrane fragments in the form of vesicles get separated along with the acid insoluble pool, and hence in the macromolecule fraction. So lipids are not strictly macromolecule. Remember this line, that lipids are not strictly macromolecules. Okay, the acid soluble pool represents roughly the cytoplasmic composition. The macromolecules from cytoplasm. Okay, the macromolecules form the macromolecules from cytoplasm and organelles become the acid insoluble fraction. So macromolecules from cytoplasm and organelles become the acid insoluble fraction. Together they represent the entire chemical composition of living tissues or organisms. So acid soluble pool represents roughly the cytoplasm, and macromolecules, they can be cytoplasm and organelles. Okay, becomes the acid insoluble fraction. So the chemical comes out okay, we observe that the water is the most abundant chemical in living organisms. Now this is the table: average composition of cells, water. These are the components, that is percentage of the total cellular mass. So water 70 to 90%, proteins 10 to 50%, carbohydrates 3%, lipids 2%, nucleic acids 5 to 7%, and ions, these are 1%. So these are the chemical composition found in the living organisms, right? Now proteins. So proteins are polypeptides. They are linear chains of amino acids linked by peptide bonds. Now each protein is a polymer of amino acids, as there are 20 types of amino acids: alanine, that is CH3, cysteine, I think sulfur, proline, tryptophan, and lysine. Lysine, it is a basic amino acid, and valine, that is neutral amino acid. A protein is a heteropolymer and not a homopolymer, right? Now heteropolymers, homopolymers have only one type of monomer repeating n number of times. So amino acids are essential for our health. So, and they must be supplied through our diet. Hence the dietary proteins are the source of essential amino acids. Therefore amino acids can be essential or non-essential. The latter are those which our body can make, while we get essential amino acids through our diet food. So proteins carry out many functions in living organisms. Some transport nutrients across cell membrane, some fight infectious organisms, that is antibiotics, some are hormones, and some are enzymes. So or collagen is the most abundant protein in animal world, and ribulose bisphosphate carboxylic oxygenase is the most abundant protein in the whole of the biosphere. Now some proteins and their functions: collagen, they act as intercellular ground substance. Trypsin is a protein, function is enzyme. Insulin is a protein, functions as hormone. So an antibody, they fights infectious agents. A receptor, they function as sensory reception, that is smell, taste and hormone. And GLUT4, that enables glucose transport into cells. So these are the some proteins and their functions: collagen, trypsin, insulin, antibody, receptor, and GLUT4. Okay, now coming to the polysaccharides. So these are the acid insoluble pellet, also has polysaccharides, that is polysaccharides, these are the carbohydrates as another class of macromolecules. So, okay, polysaccharides are long chains of sugars. Polysaccharides are carbohydrates, they are sugars, they are the threads. Okay, example are cellulose is a polysaccharide containing only one type of monosaccharide, that is glucose. Cellulose is a homopolymer, right? Starch is a variant of this cellulose but present as a storehouse of energy in plant tissues. Okay, animals have another variant called glycogen. Now inulin is a polymer of fructose. In polysaccharide chain, say glycogen, that is a storehouse present in the animal cell. The right end is called the reducing end, R for right, R for reducing, and the left end is called the non-reducing end. Now the glycogen has the branches. Now starch, which is a storehouse in the plant tissues, okay, which is a homopolymer containing the one type of monosaccharide, that is glucose. They form the helical secondary structures, and this is why they can hold I2 molecules in the helical portion. While this starch I2 is blue in color, it is a test, but the cellulose does not contain the complex helices, so hence it does not hold the I2 molecule. Now you can see the diagrammatic representation of portion of a glycogen. You can see two glucose molecules, they are dehydrated, right? 1 2 3 4 at the fourth position, and one position of two ring, as you can see. So this is it, like as you can see, it is a branched, and this one is the glycogen. Now plant cell walls are made up of cellulose, and they made to make the paper. Paper is also made from the plant pulp and cotton fiber, which is also cellulosic. Okay, there are more complex polysaccharides in nature. Okay, and they have the amino acids, amino sugars, and chemically modified sugars, that is glucosamine and N-acetylgalactosamine. Glucosamine and N-acetylgalactosamine. Now, exoskeletons of arthropods, for example, have a complex polysaccharide called chitin. Now these complex polysaccharides are mostly homopolymers, right? So exoskeleton of arthropods have a complex polysaccharide called chitin, and these complex polysaccharides are mostly homopolymers. Okay, now nucleic acids. The other type of macromolecules that one would find in the acid insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides together with polysaccharides and polypeptides. So polynucleotides, polysaccharides, and polypeptides, these comprise the true macromolecule fraction of any living tissue or cell. And for nucleic acids, the building block is a nucleotide. A nucleotide has three chemically distinct components. One is a heterocyclic compound, it can be pyrimidine or purine. The second is a monosaccharide, that is a pentose sugar, and third is a phosphoric acid or phosphate. Now the heterocyclic compounds in nucleic acids are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. So heterocyclic compounds, these are the nitrogenous bases. They are named adenine, guanine. They are the two heterocyclic ring, and uracil, cytosine, and thymine, pyrimidine. They do have the one heterocyclic ring. Okay. Now adenine and guanine are substituted purines, while the rest are substituted pyrimidines. Now the skeletal heterocyclic ring is called as purine and pyrimidine respectively. The sugar found in polynucleotides is either ribose, a monosaccharide pentose, or 2-deoxyribose. Okay, the sugar found in polynucleotides is either ribose, a monosaccharide pentose, or 2-deoxyribose. So it's not the glucose. Okay, it's pentose or 2-deoxyribose, because at the 2' position there is no OH. A nucleic acid containing deoxyribose. Now those nucleic acid that can be RNA or DNA that contains the deoxyribose type of sugar is called a deoxyribonucleic acid DNA, while that which contains ribose is called ribonucleic acid. Now structure of proteins. Proteins, as mentioned earlier, are heteropolymers containing strings of amino acids. Now structure of molecules means different things in different contents. Okay, it's not that important, but okay. We biologists describe the protein structure at 4 levels. The sequence of amino acid, that is the positional information in a protein, which is the first amino acid, which is second, and so on, is called the primary structure. So primary structure of amino acids of proteins gives the sequence of amino acid, that is the positional information in a protein. Okay, a protein is imagine as a line, the left end represented by the first amino acid and the right end represented by the last amino acid. As you can see in the first diagram, it is a primary structure of the protein. It is a polypeptide. Okay, it is just a line. The first amino acid is also called as N-terminal amino acid, and the last amino acid is also called as the C-terminal acid. Okay, the first one is the N and second and last one is the C-terminal amino acid. The protein thread does not exit throughout as extended rod. The thread is folded in the form of helix. Okay, of course only some portion of the protein thread are arranged in the form of helicals, not all. Okay, so not all present in the form of the helix. Now in proteins only right-handed helices are observed, but in protein only right-handed helices are observed. Other regions of the protein thread are folded into other forms in what is called the secondary structure. Now the other regions of the protein thread are folded into other forms in what is called the secondary structure. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to the tertiary structure. This gives a three-dimensional view of a protein. The tertiary structure is absolutely necessary for the many biological activities of the proteins, because they have the active sites. So these are the structures, that is secondary, that is alpha helix and beta plated sheet, and some of them form the tertiary, that is hydrogen bond and disulfide bond. Then the quaternary structures. So these are the various levels of protein structure. Okay, now some proteins are an assembly of more than one polypeptide or subunits. Now the manner in which these individuals folded